Good morning, students. So today I am going to deal with a new topic, that is glycolysis. This is one of the phases of respiration. As you all know, respiration is the breakdown of glucose that takes place just to extract the energy. All the cells undergo respiration because they all have mitochondria. But there are some exceptional cells that does not have mitochondria, like RBC cells. So they don't have mitochondria, so they may undergo anaerobic respiration. And even in the case of what we say muscle cells, they may undergo respiration, that is anaerobic. When you do strenuous exercise or heavy exercise, your what is called the muscle cells, they undergo the respiration which is anaerobic and which produces the lactic acid. But in plants, in case of anaerobic respiration, they produce ethyl alcohol when there is incomplete breakdown of glucose. So right now we are talking about the glycolysis. So there are 10 steps taken place in the glycolysis. But the basic concept according to your syllabus, only you have to understand is glycolysis is the first step of respiration process, okay, and the second step is called Krebs cycle. So in the glycolysis, it takes place in cytoplasm. It takes place in cytoplasm, right? You have a cell here, okay, inside our human body. There are numerous of cells. So here inside the cell, you have mitochondria. So inside the mitochondria, what we know, that Krebs cycle takes place, but the glycolysis takes place outside the mitochondria, okay. So that will be in the cytoplasm of the cell. So this glycolysis, it is the common phase of aerobic and anaerobic respiration. So there are several steps taking place in the glycolysis. In glycolysis, there are 10 steps taking place, okay. So first concept, what is to be done in the glycolysis is that in glycolysis, glucose is converted into pyruvate, that is 2 molecules of pyruvate. So this step is called glycolysis, and between these steps there are 10 steps taking place, okay. 5 steps is for investment phase, then another 5 steps is for just energy production, okay, that is payoff phase we call. So before understanding this glycolysis, first you have to know where is the respiration taking place, in which cell, how is the procedure taken place. Example, you have a stomach right now here, then duodenum, which is connected, which is the first part of the small intestine, is connected to the large intestine, as you know already. Yes. So here in the hepatic portal system, I have explained that the blood which has been travelled from the intestines or from stomach, this vein will join as the reunited and join as the hepatic portal vein, okay, hepatic portal vein. So this hepatic portal vein, now it enters the liver, this blood, before going to the circulatory part, okay. Circulation means heart and blood vessels. So when the blood is enters to hepatic portal vein to the liver, the blood containing glucose, this glucose needs to be broken down for the entire respiration of the cells. So this glucose, then how the main thing is that how the cell will get the glucose. The question is how the cell will get the glucose. So to get the glucose from the cell, the first process, because the first process in glycolysis is glucose, So, glucose has to be converted, has to be split into two molecules which ultimately forms pyruvate. Okay. So, first process is that they need glucose to be entered. Okay. So, before entering the glucose has to first along with the blood enters to hepatic portal vein and it enters into the liver. Then once the liver cells, they have certain what we call hepatocyte cells. Okay. They are called hepatocyte cells. Here the veins which has been reunited as the hepatic portal vein, they will break down into capillaries, okay, into capillaries and again reunited to form hepatic vein as we have studied in the circulatory system. So, this hepatocyte cells are here around. So, this capillary surrounds all these hepatocyte cells. Now, the function of liver is that when the glucose in the blood enters the liver, the glucose which is excess, they will be what we call converted into glycogen, means so that they can be stored in the liver. So, this excess glucose will be converted into glycogen and the remaining glucose which has to be transported, they will be entering the circulatory system, that is heart and blood vessels. Then finally through the capillaries they will leak out into the tissue fluids. So, when they enter into the tissues which has been leaked out from the capillaries, now the cell needs respiration. So, cell needs always respiring because they need to be respiring because they need oxygen. So, whenever they do any activity, they need oxygen. So, whenever they do the activity, oxygen, the blood becomes again deoxygenated. So, the oxygen has to be added up again and again. So, once the circulatory part they have entered, now it has entered the cell. But to enter the cell, they need the glucose to enter the cell, they need certain transporters they need, okay, certain glucose transporters, okay. There are types of glucose transporters. So, these glucose transporters can be produced from the cell. The cell has what? Inside the cell what? There is nucleus. So, here nucleus, they consist of nucleic acids, DNA, RNA. So, this RNA may send as messenger RNA, then this messenger RNA will help in translating the proteins, producing the proteins. So, these proteins, they are called glucose transporters. So, they will form the glucose transporters along the membrane. So, this is the way how glucose enters the cytoplasm. So, now you understood how the glucose has entered the cell. Now, the glucose is available inside the cell. So, once the glucose is available inside the cell, now glycolysis can take place. Now, let us see what is the process taking place over here. So, let us have an overview of the glycolysis, the steps. So, the first starting compound, that is called glucose. Okay. So, this glucose, what is the main role of glucose? That is to break down. So, to break down, now what is the process in the first step? For any business when you invest some money, then you get more money. So, same way in this also, to produce more energy, I am going to invest more of ATP. As you have studied the ATP cycle, right? In ATP cycle, how the energy has been produced and how the energy has been stored in the form of ATP. So, this ATP Okay. So glucose here, it will be converted into glucose 6 phosphate. Now understand this first step, that is by the addition of the phosphate, because I have invested 1 ATP, okay, by the removal of phosphate. So here I have removed the phosphate from ATP and added to the glucose. So 1 phosphate has been removed from the ATP. Adenosine triphosphate means 3 phosphates are present, okay. Then adenosine diphosphate means 2 phosphates are produced, because 1 phosphate has been already removed for the attaching to the glucose. So this phosphate, now this is called phosphorylation process. Phosphorylation, because I have added 1 phosphate in the glucose, okay. So in the glucose ring, as you all know, glucose ring is somewhat like this, and you will see that here okay, isomerase enzyme, this glucose 6 phosphate will be changed to fructose 6 phosphate. That is just a rearrangement process, just a rearrangement process. That is, they have the same chemical formula, but stereochemical arrangement of atoms will be different, okay. Because here in the fructose you will see just like this, like pentagon, okay. Here hexagon, here pentagon, like that. So oxygen here, then here 1 carbon atom, here 2, then 3, 4, then 5, then 6 here, okay. So what is the purpose of I am isomerizing the glucose 6 phosphate to fructose 6 phosphate? Because one more phosphate we need to add here, okay. So what is going on inside the cell? So 1 phosphate already added here, 1 phosphate is already added here, then another phosphate I need to add. So that is at the first place. So now you see here, if the glucose 6 phosphate has been used, we cannot add the phosphate here. So that's why they have isomerase to fructose 6 phosphate and 2 phosphates now added. 1 phosphate is at first place of the carbon atom, here first place, this is first place, then here 6th place the phosphate is added here. So this is the next step. In the first enzyme, if you talk about the enzymes, now these enzymes all come from where? All these enzymes, these all have been produced there itself inside the cytoplasm, okay. They are already present in the cell. So all these respective enzymes help in the conversion of the chemical compounds. So here which enzyme it is used? That is hexokinase. Hexokinase enzyme, because it is of 6 carbon atom. So here also it is 6 carbon atom. Glucose 6 phosphate, there is no change in the carbon atom, 6 carbon atom, only 1 addition of phosphate has taken place, and here only just rearrangement of the structural type of the carbon atoms, but the phosphate but the carbon atom will be same number of carbon atom, okay. Now what is the next step here? In the next step, here phosphorylation has been done. In the first step, phosphorylation means anything, the phosphate which is added to the compound, okay. So in the first step, this is phosphorylation. Now the second process again, that is phosphorylation is going to happen here, because I am going to add this phosphate here, phosphate here. So fructose 1,6 biphosphate or diphosphate, okay. So this is also 6 carbon atom, this is also 6 carbon atom. Now with the help of phosphofructo enzyme, okay, with the help of phosphofructokinase, it has formed the fructose 1,6 biphosphate. Now let us move on to the next step. Here, before moving into the next step, this step, okay, this step is investment phase, okay. This step is called investment phase. Investment phase means I have invested one ATP here, and here one more ATP will be invested, okay. One more ATP will be invested here. So, ATP 1-phosphate is utilized again here. So, up to here you must understand what is going on. Now, the next process is just splitting this fructose 1,6 biphosphate into two chemical compounds having the 3 carbon atom each, okay. So, now let us see here what happens here. So, in the fructose 1,6 biphosphate, with the help of aldolase enzyme, so all the enzyme names I will be writing with the blue color. So, aldolase enzyme it has been converted into dihydroxyacetone phosphate, and now the compound formed is glyceraldehyde 3-phosphate now after splitting it. So, I have repeated twice because they are interconvertible to each other, isomer of each other, okay. So, more focus going towards the glyceraldehyde 3-phosphate. So, this glyceraldehyde 3-phosphate will be converted into 1,3-biphosphoglyceric acid. So, before that you need to understand is that hydrocarbons, when they undergo oxidation, they produce hydroxide compound. Then when hydroxide undergoes the oxidation purpose, then they produce aldehyde group. Then when aldehyde group of compound, when they undergo oxidize, when they are oxidized, they form what we call, that is carboxylic acid, means acids are formed from the aldehyde group. And when carboxylic acid undergoes oxidation, it produces carbon dioxide. So, this process you may be knowing about it. So, this I want to show is that this one aldehyde that changes into glyceraldehyde, okay. Now, here during this step, dehydrogenase enzyme, okay, which is present in the cytoplasm, this dehydrogenase will help to remove the hydrogen atoms. Now, the hydrogen has been removed, okay. Whenever there is dehydrogenase process, the hydrogen will be removed. So, 2 hydrogen atoms has been removed. Now, these 2 hydrogen atoms will be picked up by NAD plus. NAD means nicotinamide adenine diphosphate nucleonide. So, this what we call nucleotide, adenine dinucleotide, this is one of the coenzyme. So, this coenzyme will pick up this hydrogen, 2 hydrogen ions. So, what does it form now? That is NADH plus plus H plus, but you will not write as what we call, usually in the book you will see NADH2. So, that you will not write, it is because it is released in the form of ion, okay, not as a compound. So, here NADH and H plus has been formed during dehydrogenase process, okay. So, whenever here hydrogen is removed, this is picked up by the NAD, which is a coenzyme present in the cytoplasm. This will form as now NADH and H plus. Now, this NADH plays a very great role. This NADH will help in producing more energy. That is, this NADH after traveling into the electron transport chain, they produce that is 3 ATPs. Okay. So, this we will talk later when I after teaching the Krebs cycle. Now, the focus is the NADH has been produced here. Now, what happens after this process, after dehydrogenase process with the help of dehydrogenase enzyme. So, this is enzyme. So, I will be putting here bracket box. Now, so now the resultant compound from this 2G3P, what will become now? Glyceric acid, because I have said aldehyde group, this is aldehyde group, glyceraldehyde 3-phosphate. So, this glyceraldehyde 3-phosphate, when it undergoes oxidation, it produces acid, and you have already studied that in oxidation and reduction there is a difference. In oxidation, what happens? That is removal of hydrogen, and oxygen gain here, right? Oxygen will gain. So, when hydrogen is removed, oxygen will gain. And in the reduction process, when hydrogen is gain, when hydrogen added gain, then what happens here? The oxygen loss. Okay. So, what happens here in this case? The hydrogen is added. Okay. This hydrogen which has been removed from the what we call compound, now it has been picked up by the nicotinamide adenine dinucleotide phosphate. Okay. So, this has been now utilized. So, this is what we call reducing process. Okay. So, this is reduction process we call. So, when it undergoes reduction process, that means NAD plus is reduced to NADH plus plus H plus. Okay. So, in the reduction, hydrogen gain. Okay. Now, the resultant, with the help of dehydrogenase enzyme, it has changed the G3P into 1,3-biphospho-glyceric acid. Okay. So, this biphosphoglyceric acid, how it is formed? One more phosphate has been added, means 3 carbon atom here. Here phosphate 3rd place, it was already added here as G3P. In the second process, by phosphorylation process, here the phosphate will be added, that is in the first carbon atom. So, that is by the process of phosphorylation. So, that we call it phosphorylation. So, this phosphorylation means what? Addition of phosphate. From where? Here we did not invest any ATP. It is from the compound, from this compound, that is mainly the compound containing H3PO4. Okay. So, from here I got the phosphate. So, now the carbon here 6, it was 6 phosphate. Now, in the 3rd place it was added. Now, in the 1st place also phosphate will be added. So, now it has formed 1,3-biphospho-glyceric acid. So, 2 phosphates have been added here, that is 1 phosphate has been added before, this one by phosphorylation process again it has been added. Okay. Now the next process, what happens? After this, 1 phosphate will be taken from the compound and will transfer to ADP to form ATP with the help of a kinase enzyme, with the help of one enzyme that we call phosphoglycerate kinase. Phosphoglycerate kinase. So, this phosphoglycerate kinase will help to pick up the phosphate from this compound to transfer to ADP to transfer to ADP. Now the phosphate has been added here with the help of enzyme, and now the ATP has been formed. So this ATP will always consider how many? 2 molecules of ATP, because from the beginning itself I have said there are 2 molecules of G3P. So everywhere you have to add here 2, here also 2, then 2 ATP, 2 ADP. So this is the first step where the ATP has been produced after the investment phase. So here ATP, the first step, first ATP production. Now what it has formed now? One phosphate has been removed and added to the ATP to form ATP, added to ADP to form ATP. Now the compound, which compound will form now? I have removed, so now it will form only 3-phosphoglyceric acid. Simple. So 3-PGA. Now this 3-PGA again will mutase, okay, with the help of mutase enzyme, that is phosphoglycerate mutase, it will be converted into 2-phosphoglyceric acid. Just the phosphate which has been in the third carbon atom has been shifted to the second carbon atom. So that's why it is a 2-PGA. Okay, but here don't confuse here. Here it is 2 molecules, here also 2 molecules, because everywhere you have to add the steps of dihydroxyacetone phosphate. Now 2-PGA has been formed with the help of mutase enzyme. Now what is next step then? The next step is just with the help of enolase, enzyme, what happens next? That is phosphoenol PEP, that is phosphoenol pyruvic acid. Phosphoenol pyruvic acid is formed here right now. Here between PGA and PEP, here what happens? They have undergone the dehydration. Okay, they undergoes the dehydration. So when they undergo dehydration, the water will be released. So water is removed out of the compound. So water is produced here. So in this case also you will write 2 molecules of water, because everywhere it is 2 repeated twice. Then this phosphoenol pyruvate also 2. Okay. So this has to be kept in mind. So all these are what we call 3 carbon atom. Here also 3 carbon atom, 3 carbon atom, here 3 carbon atom, till it finishes the last step, that is pyruvate, okay, is formed. So this pyruvate is formed by dephosphorylation. Okay, here dephosphorylation takes place, because one phosphate is what removed from the compound to add to the ADP to form ATP, and that is called dephosphorylation. So phosphorylation and dephosphorylation don't have a confusion here. Here it is ATP to ADP, that's called phosphorylation. Means the phosphorylation, the phosphate has been added here, but here it was rain, so I stopped here. Now I will be continue. So just a little bit end is left, that is last step, that which I was saying, that pyruvate is produced here at the end. Okay. Now you see here the whole process of glycolysis. The first starter is glucose. Now glucose has been converted into 2 molecules of pyruvate. Okay. So since the whiteboard is completely wet now after the rain, so I am unable to write now. So I will be writing it again. 2 pyruvate 2 molecules of pyruvate is produced at the ending stage of the glycolysis. Okay. So, this now the whole process, let us discuss again. Overall view of the glycolysis will be just the first state, that is glucose is needed in the cell. This glucose will be converted into glucose 6 phosphate by the addition of the 1 phosphate at the 6th carbon with the help of exokinase. So, here phosphorylation happens because addition of phosphate happens to the compound. So, phosphate is added. So, it is called phosphorylation. So, ATP is invested here, 1 ATP. Now, glucose 6 phosphate will be isomerized to fructose 6 phosphate with the rearrangement. Only the chemical formula will be same, only the stereotype chemical arrangement of atoms will be different. So, as I have said here this is the chemical arrangement of the glucose 6 phosphate and here the arrangement of fructose 6 phosphate. So, that there will be 2 end. Okay. Here side ring is there, 1 ring, here second side ring. Here only 1 side ring is there. So, we cannot add the another phosphate here, but in the fructose 6 phosphate we are adding the another phosphate here. So, I have to change this glucose 6 phosphate to fructose 6 phosphate. So, they are isomers of each other. Okay. Now, the phosphate is added here at the 6th and the 1st place. Now, fructose 6 phosphate will be again converted into fructose 1,6 biphosphate by the addition of 1 more phosphate at the 1st carbon atom. So, it has now with the help of phosphofructokinase. So, these certain enzymes are present in the already in the cytoplasm. So, these enzymes will be helpful in conversion of these compounds. Now, fructose 1,6 biphosphate, it has been again now split into 3 carbon atom, that is dihydroxyacetone phosphate and glyceraldehyde phosphate. So, this is a DHAP is the what we call acetone group, what we call that ketone group, and this is aldehyde group. So, ketone and aldehyde groups these are interconvertible to each other, they are isomer of each other. So, the more they are equilibrium to each other. So, the more focus we are giving towards the glyceraldehyde phosphate, because the same chemical compounds will be formed from here as well as from here too. So, we are just repeating the twice the formation of the compounds. So, now here you have seen the 2G3P. So, this we are utilizing instead of DHAP. Now, G3P, that is glyceraldehyde 3 phosphate, phosphate which is at the 3rd carbon atom. So, this is always a 3 carbon atom because it has been split from 6 carbon atom. Now, I have repeated it twice because instead of writing all these compounds which is formed from DHAP, we are just repeating it twice here. So, I have written here 2 molecules of G3P. Now, with the help of dehydrogenase, dehydrogenase means hydrogen will be removed from the compound. So, hydrogen will be removed from the compound in the sense I have said that any aldehyde group, when on oxidation the hydrogen is removed, then acids will be formed. So, here as I have discussed earlier when I was explaining this process. So, this under dehydrogenation, the hydrogen has been removed from the compound. Now, it has been picked up by the NAD to form NADH plus H plus. So, this also will be 2 molecules, 2 NADH will be produced here. Now, the next step is that 1,3-biphosphoglyceric acid. So, this 1,3-biphosphoglyceric acid, already the phosphate was present at the 3rd place. Now, here another phosphate I have to add in the 1st carbon atom. Okay. So, in that case, what under this phosphorylation process, 1 more phosphate is added. Okay. Now, the next step. So, all these are because of the enzymes. There are certain enzymes to help in conversion of this. Now, 1,3-biphosphoglyceric acid has been produced now. Now, the 1-phosphate, that is dephosphorylation. Here in the first step, dephosphorylation will take place here. Phosphate will be removed from the compound and this phosphate will be transferred to ADP to join with the ADP to form ATP. So, this is the first step where ATP is produced here. So, first ATP is produced, so but I will count it as 2 molecules of ATP because from this 2 compounds. Now, this 3 PGA will be again what we call the phosphoglyceric acid. I have said that from aldehyde group has been converted to glyceric acid because of the dehydrogenase enzyme. Okay, means the aldehyde always change to acid when it undergoes the dehydrogenation process, means when it is undergoing oxidation process. So, oxidation of G3P has taken place, aldehyde group, and it has formed acid now, and here also the same acid is formed, only the phosphate, this one phosphate will be removed from the compound and it will be transferred to the ADP to form ATP. So, ATP is produced here. Now, only 3 PGA, means the phosphate only one is present at the third carbon atom. So, that is why it is a 3. So, 3 PGA has been produced. Now, 3 PGA is and 2 PGA are isomer of each other. They have same chemical formula, only the stereotypic chemical arrangement of the atoms will be different. So, here 3 PGA, here 2 PGA, so just carbon rearrangement. Here the phosphate is at the third place of the carbon atom, and here the phosphate has been shifted to second carbon atom. So, finally 2 PGA is formed. Now, this is with the help of enzyme mutase. Now, in the phosphoglyceric acid will be converted into phosphoenol pyruvate, that is with the help of enolase enzyme. Here undergoes dehydration. Okay, here it undergoes dehydration. So, dehydration means what? Water is removed. So, that is why here I have shown water is removed. Those 2 molecules always everywhere. Then, because repeated twice, the next is phosphoenol pyruvate is formed. This phosphoenol enol pyruvate is, once it is formed, this is also 3 carbon atom, and this will finally it will form 2 molecules of pyruvate acid by the formation of ATP by the dephosphorylation process. Dephosphorylation means the phosphate will be removed from this compound and will be transferred again to the second stage, okay, added to the ADP to form ATP. Means here first step ATP produced, next second ATP produced. So, finally pyruvate molecules are produced here. Okay, fine. So, these are the certain steps. This is an overview of all the steps of glycolysis. Now, I have said that this first 5 steps they are called investment phase, and here this another continuing 5 steps they are called payoff phase. Okay. So, in the payoff phase you can see here the 2 ATP is produced and 2 ATP is produced here. Okay. So, 4 ATP are added, and in the case, so I am adding here as 2 ATP and here 2 ATP. Clear? Now, NADH which has been produced 2 here, when it is plays a lot of water production in energy. Okay, so it produces energy more when, so not now, but it has to enter the electron transport chain. So, after undergoing the electron transport chain, it comes back to here. So, that process I will explain the Krebs cycle while I am teaching the Krebs cycle and glycolysis together. So, here each NADH, always keep in mind that there will be 3 ATPs produced. Okay, always this you keep in mind. One NADH will produce always 3 ATP produced. Now, this NADH will be produced three. So, here it is 2. So, 3 2s are 6. So, 6 are produced here, that is 6 ATP. So, here I am writing 2 NADPH produces what we call 6 ATP. Fine. Now, let me rub this so that I can add it. So, this 6 ATP which is produced from NADPH, how many ATPs are produced? 10 ATP. Fine. Now, this 10 ATP you minus it from 2 ATP, because 2 ATP it was just invested as a business purpose, like to produce more energy. So, 2 ATP is produced. So, we can see here out of 4 ATP produced here, 2 2 ATP we have gained, means 2 already we have invested, so 2 have gained. Now, so 2 will remove it, so 8 ATP has been produced totally in the glycolysis state. Okay. Now, finally, the overall reaction in glycolysis, okay, the overall reaction in glycolysis process is that glucose, okay, here the glucose is produced and 2 ADP has been utilized here, 2 ADP has been utilized, and then what else, 2 NAD has been also utilized, and also inorganic phosphate, that is phosphate has been added, phosphorylation process, 2 process, so 2 inorganic phosphate. Now, the products formed, that will be 2 ATP produced, because 2 ATP are gained here. The 2 ATP investment we have invested here, 4 ATP has been produced, but 2 ATP is the gain. So, it will be taken as 2 ATP. Then NAD, which is reduced, that is called reducing agent. So, in the NAD which has been reduced to NADH, that is 2 molecules. Okay, here also 2 it will come. Then next is 2 ATPs, or ATP I have given already. So, 2 NADH, then what else is left behind, that is H plus, okay, hydroxide ions. So, 2 hydrogen ions has been produced, and water which has been removed from here, water is released. So, water also has been formed, 2 molecules of water. So, this is the overall reaction which takes place in the glycolysis. So, I hope you have understood what is glycolysis, and now next we will go back to Krebs cycle. Okay. So, this is one of the phase now, this is just the first step of the respiration process. Now, second step of respiration process we will discuss in the next class. So, thank you for today's class. Let us